experiencing a revolution in the sense of how cancer treatment is approached these days. We realize that the immune system can really be a very strong barrier against cancer. And the so-called immunotherapies are really inducing massive responses across different tumor types. The problem is that not all patients respond. Actually, there's a reduced fraction of them that derive long-term benefit. So there's a huge amount of interest in understanding why that is the case and what are really the bases that led to some patients to respond or not. And what we basically study is that one of the reasons why patients respond is because the tumors are already inflamed when you treat them. And by that I mean that they are already infiltrated by a specific type of immune cells that make those tumors respond to those treatments. However, there's also another type of inflammation, which often happens in patients, which actually promotes tumor growth and resistant to therapy. So there's a type of inflammation that we so-called good, that is associated with the responses to the treatments, and another one that is bad, that is associated with resistance. And we try to understand what leads to this interaction and how we can actually manipulate the inflammatory flavor within a tumor. So in this particular study, we actually found that the use of anti-inflammatory drugs that are commonly used for treating conditions such as arthritis can actually help and boost the response to those immunotherapies. So rather than really generally stopping the inflammatory response within the tumor, they seem to sort of favor a type of inflammation that helps those immunotherapies. What we found is that actually all anti-inflammatory drugs that target a pathway known as cyclooxygenase 2 prostaglandin E2, which is very often expressed in tumors, seem to induce this response, including drugs such as corticosteroids, which are widely known as immunosuppressive drugs. So quite surprising, what we found is that corticosteroids that are used to treat the toxicities that are often associated with these treatments can actually boost the response in our specific experimental settings. So it remains to be tested to what extent, in which settings, this might be applicable in cancer patients.
Well, actually, what remains surprising to a lot of us is the fact that in mouse models that are genetically the same, eat the same food and carry the same tumors, they can still show completely opposing responses to these treatments that we've just talked about. Some mice fully reject the tumors and others succumb to tumor progression. So this is actually quite similar to what we see in patients, but it does make the identification of the really early mechanisms of response more complex because of this whole mixed bag that we see. So to overcome this, we designed a surgical model where we implanted 2 tumors into the mice instead of 1, and we surgically removed one of them early on when we thought that perhaps the protective mechanisms were at play. This meant that we were able to then look at the mechanisms of response in tumors in mice that we knew for certain went on to respond well to treatment. So this was a game changer in terms of identifying really the mechanisms in place to drive a response in the combination treatments.
So we think that maybe one of the most surprising results was that our work in mouse models demonstrated that a single dose of anti-inflammatory drugs, which are commonly used to treat chronic inflammatory diseases, can actually rapidly and acutely shift the inflammatory profile within the tumors, making them more responsive to the immunotherapies. And interestingly, this molecular change could actually be achieved by using new drugs that selectively stop the action of prostaglandin E2 on the immune cells, making them more fit to kill the cancer. So overall, we think that our findings establishes this prostaglandin E2 pathway as an independent immune checkpoint that can readily be targeted in cancer patients.
Yeah, we were very lucky to collaborate with Daniela Thoman and Ton Schumacher at the Netherlands Cancer Institute, who have essentially developed a game-changing platform to test responses to immunotherapy in a dish outside the human body. What they showed is that in tumor fragments that they dissected following the surgical removal of tumors, they could essentially predict responses to immunotherapy in those patients within a 48-hour period. So I received a travel award to spend a summer in their lab and test the effect of anti-inflammatory drugs in that setting on the immune profile of human tumors. And we found that similar to what Agren was describing in the mouse models, we found a rapid shift in the inflammatory environment of the human tumors towards one that is known to be good for recruiting the protective immune cells and generally good for responses to immunotherapy. So it's a really incredible tool to test new compounds, and I encourage everyone to read their study and ours to learn more about it.
The study has many implications, but one of obvious clinical implications is this idea that you could actually treat patients that have been treated with immunotherapies and add to that these widely used anti-inflammatory drugs to see whether they enhance a response. So in fact, us here in Manchester and others in the world are actually testing now in clinical trials whether the combination of anti-inflammatory drugs and immunotherapy can really boost the response. I think this is a very key next step, and hopefully in the next years we'll know whether this is really something that can really help and boost the response to this treatment.